Welcome to EduRev.
Welcome students, I'm Callus Culture and I have been training NEET medical aspirants in botany as well as in zoology and also NCERT based board exams for the past 8 years. In the last video we have already studied a little bit about reproduction in organisms. So this will be the second class on reproduction in organisms. So let's have an overview of what we're going to study today. In the previous video lecture, we have already studied some of the asexual reproduction types, and today we will continue studying the remaining types of asexual reproduction. Now, just to have a small overview, in the last class we have finished fission, budding, regeneration, and fragmentation. Today we will complete sporulation and vegetative propagation as well as Pathanogenesis. Now, coming to sporulation or spore formation. Sporulation or spore formation is very common in kingdom fungi and in lower plants, for example, in case of algae. Now, spores are of different types. They can be motile as well as non-motile. For example, if they are motile, they are called as zoospores, whereas if they are non-motile, they are called as planospores. Now, why the term zoospore and planospore? It's very easy to understand the word zoospore simply because the word zoo comes from the word zoology, implying animal-like, which means most probably these are going to be motile, they can move. Okay. Now, motile spores are often defined with the help of flagella, and generally speaking, they are biflagellated, for example, in case of Chlamydomonas. However, non-motile spores are non-flagellated, therefore they cannot move. Now, a couple of examples are seen here in case of Penicillium and in case of Rhizopus. So let's have a brief go through with both these two types of spores. Now the first one being zoospores. So if you can look at the zoospore figure that we have over here of Chlamydomonas, you will find that it is biflagellated. It looks very similar to the unicellular organism Euglena. Okay, but this particular biflagellated structure, you need to understand that the presence of flagella itself should tell you that Chlamydomonas probably requires an aquatic medium for its reproduction. Now, what we really need to focus on are the type of planospores. Generally speaking, there are two types of planospores that we study. One is called as conidia, the other one is called as sporangiospores. Now we need to understand what are the differences between conidia and sporangiospores. So first we start with conidia. Now if you look at the figure of conidia, conidia is a type of spore that is often seen in kingdom fungi. Now I'm sure you have studied the different parts of fungus being hyphae, mycelium, etc. Okay, so if you look at the structure of this particular fungus, this is actually the hyphae that you can see over here. These protruding structures are hyphae. But definitely these hyphae are very different from what you have studied in school. So this is a very specialized type of hyphae or very specialized type of hyphae called as conidiophore. And on this particular conidiophore, you can see a couple of spores being formed on the external surface. So they are pulled out externally. So can we say that conidia, which are these particular spores, are exogenously produced? Okay, so if you look at conidia, first of all, exogenously produced, and they're produced on specialized hyphae called as conidiophores. Now, coming to the second type of planospore. Now this particular figure looks very similar to what we have probably studied in our school. So here you have sporangiospores in Rhizopus. So Rhizopus is nothing but our bread mold. Now here you can find the presence of sporangiospores present within a capsule, okay, a capsule called as the sporangium. And this particular sporangium is present on hyphae called as sporangiophore. So in the previous case, we had the presence of conidiophore, but now we have the presence of sporangiophore. So can we say that these particular spores are formed within an encapsulated structure? So they're formed inside the sporangium. So they are endogenously produced and they're produced on certain type of hyphae called as sporangiophores. So we need to remember these differences. Okay. Conidia are exogenously produced, whereas when we speak about sporangiospores, they are endogenously produced. This is the characteristic difference between the two planospores. Now, coming to the next type of asexual reproduction, and that is vegetative propagation. Vegetative propagation is normally seen in various natural occurrences. Okay, so you have natural vegetative propagation and you have artificial vegetative propagation. Now, natural vegetative propagation you are very familiar with. For example, the eyes of the potato, rhizomes in case of banana. Okay, you've studied this from your school level. But what we're going to focus on is artificial vegetative propagation, which is often used in horticulture. Now, actually speaking, there are four types of artificial vegetative propagation, that is grafting, layering, cutting, and micropropagation. But out of these four, you will find that two of them are really important for the NEET exam, and that is grafting and micropropagation. So we are going to focus mainly on these two. Now, starting with grafting. Now, in order to understand grafting, let's take a simple example. Let's say we have two plants. In the first plant, you have a very good root system, an extensive root system, and this particular plant is able to absorb water as well as mineral nutrients very, very well. But for some particular reason, this particular plant, okay, as it starts sending its nutrients upwards towards the shoot system, probably the fruiting of this particular shoot system is not very extensive, it's not a lot, or maybe the fruits are not very tasty, but the root system of the first plant is very, very good. Now let's consider another plant. In this particular second plant, the root system is very weak. However, this particular plant at the shoot system gives you very good fruit, probably very tasty fruit. Okay, so now we've got two plants in which one has a good root system, whereas the other one has a good shoot system. Now, grafting is a procedure by which I can bring the two good parts of these plants together. Now, how do we do that? We take the extensive root system and we are going to call this as the stock. We're going to take only the root system of the first plant. And we're going to cut off the shoot system from the second plant and we're going to take that shoot system and we're going to call it as the scion. Now we're going to join these two, the root system of the first and the shoot system of the second. So the stalk and the scion are going to be joined together. Okay, and after some time we're going to propagate these two as one individual plant. So in other words, what have we done in grafting? This new plant that will come out will have a good root system. It's going to pull up a lot of water, a lot of mineral nutrients, and it's going to supply it to a shoot system that's able to produce extensive, good and tasty fruit. Okay, so this exactly is what grafting is. So if we go through the technical definition, grafting is a technique of connecting two parts, usually a root system and a shoot system of two different plants, in such a way that they unite and later develop as one composite plant. So the whole idea is join these two together to get one really good plant. So just for our memory's sake, which is really important for us, the shoot is called as the scion, whereas the root is called as the stalk. Now coming to the second part, that is micropropagation, also called as plant tissue culture. Now if you look at the term micropropagation, what does the word micropropagation really mean? It means propagating a full plant using something that is micro, using something that is small. For example, if you go through the steps of micropropagation, you will understand that we make use of very, very small plant tissue material, and from that small amount of material, I am able to produce one extensive big plant. Now, how is this really done? Now, let's go to the definition first. Okay, so it is a propagation of large number of plants during a very short period. Now, if you know, if I try to cultivate a plant, it's going to take me a lot of time because of growing season. Okay, but in terms of micropropagation, I'm able to do that. I'm able to have extensive cultivation very, very quickly. Okay, so large number of plants in a short duration. This method of producing thousands of plants using tissue culture is called micropropagation. Now let's go into the understanding. Now plant tissue culture micropropagation has 5 steps. Let's go through them. Collection of something called as the explant, growing of the explant to produce a callus, so we'll understand later what's a callus, separation of single cells of this particular callus, propagating the cells into a plant, and finally acclimatization of those particular plantlets to the field. Now let's go into them one by one. The first is collection of an explant. Now what do we understand by an explant? Say suppose if you've got a particular plant that has really good properties and I want to create multiple plantlets from it. Now an explant is any small tissue taken from this particular good plant that we already have growing. Okay, now this particular explant, once removed, must be maintained in sterile conditions, and you must be able to grow it in a test tube or maybe say in a petri plate using good nutrient media. Okay, so an explant is defined as it's any part taken from the plant and grown in a test tube under sterile conditions in special nutrient medium. Now let's look at a practical example here. We can probably take maybe say a piece of the bud. We can take a leaf, a small portion of a leaf as the explant. We can take a small portion of the stem for that matter, or maybe even a small portion of the root. So any part can behave as an explant tissue sample, and I can grow this either in a petri plate or I can grow it in a test tube. Now let's go on to the second step. Growth of this particular explant to produce a callus culture. Now, in order to understand what's the meaning of the word callus culture, let us first understand what is the nutrient medium that we're growing these particular explants in. Now, the nutrient medium obviously has to support growth, so you require a carbon source. Generally speaking, the carbon source is sucrose that is used. You can use other carbon sources. But along with that, remember we are propagating plants, so you require a whole lot of micronutrients. If you remember, plants need lots of minerals, so you have to add these inorganic salts in. To that, you will add vitamins, amino acids, anything that is essential for growth. But what's most important is, in this particular explant nutrient medium, we have to add proper concentrations of growth regulators, especially your auxins and your cytokinins. Now, why is that so? Depending upon the ratio of auxins and cytokinins, you will find that you're able to convert this particular explant, which is a mature tissue. Remember, if you're part of the leaf, stem, or root, you have already become mature, implying you are differentiated. That's the technical term. You're mature. But the moment you apply auxins and cytokinins in particular ratios, you can take that differentiated mature tissue and convert it into a completely undifferentiated immature tissue. Okay, so that is our understanding here. And the moment we convert it into an undifferentiated tissue, okay, immature tissue, this undifferentiated mass is going to be called as the callus. So dedifferentiation or undifferentiation of the explant. Okay, so you have undifferentiated cells, and these undifferentiated cells are called totipotent. Now, what's the meaning of totipotent? If I take a permanent cell and if I transfer it back into an immature cell, so mature into immature, this immature cell now is like a stem cell. It has total potential. Totipotent means total potential to form anything, which means it is behaving as good as the zygote. Now I want you to imagine this. We had taken a small piece of the leaf and we have grown it in this particular nutrient medium, but now this whole leaf, the small portion of the leaf that I've taken, has become a callus culture, which means it has become completely undifferentiated. Now the advantage here is this. Each cell of this particular callus is behaving as an individual zygote, okay, which means all of them have the potential to give rise to new plants, each cell. So the next step is this. Now from that big mass of callus, separate single cells from the callus culture. So here is what we do. We take this particular callus, put it probably into a beaker and separate them into single cells. Now what is the advantage behind this? Each cell can be propagated into a new plantlet. So if you look at step number 4, use those cells, propagate them into individual plants. So here you can see in the last part of the figure that each cell will give rise to a single plantlet. But now I want you to understand this. Where have we conducted all these particular experiments? All of them have been conducted in the laboratory, which means we have given best possible conditions. Now we need to be able to transfer this particular plantlet into the field. Now that's not very easy because this plantlet has grown under good conditions. So therefore, in the last step, acclimatize these plantlets to the field. How do you acclimatize a plantlet to a field? Allow to grow in a greenhouse, okay, and over there you keep on removing the good conditions that you've given it. Maybe for a couple of weeks, remove supplying ample amount of water. After some time, do not supply it good amount of nutrients, and finally you will get this particular plant ready for the field. So that is acclimatization. So students, these are the 5 steps involved in micropropagation. Now micropropagation, the new plantlets that are formed are going to be termed as soma clones. Now why do we call them as soma clones? You are already aware that when we speak about asexual reproduction, the young ones are called as clones. Now, when we pay attention to micropropagation, okay, you have realized that we have taken the somatic cells of the plant, okay, and not the gametic cells, and therefore the new individuals that arise from micropropagation specifically come in from the somatic cells, therefore called soma clones. Okay, so here each of these plants are genetically identical to the original plant from which they were grown. That is the whole idea behind formation of clones or sexual reproduction. Now, commercially, we grow a number of plants using micropropagation. For example, banana, apple, tomato. Okay, a lot of plants are commercially grown in this particular way. Now, let's try to understand something a little bit more about asexual reproduction. If you remember, in the beginning, we spoke about asexual reproduction as formation of offspring from a single parent, but we left the second part of the definition which stated with or without the involvement of gamete formation, which means sometimes asexual reproduction will take place with the formation of a gamete. Now, all the types of asexual reproduction that we have studied did not involve gamete formation. However, now we come across a particular type of asexual reproduction called Pathanogenesis, which requires the formation of gametes. So here it is development of offspring from the gamete. However, the gamete does not undergo fertilization. So development of offspring without fertilization. That is, gametes are formed but gametes do not fuse. Now let's take an example of Pathanogenesis. Pathanogenesis is very often or very easily understood and seen in case of honeybees. Okay, so if you are well versed with honeybees, there are a variety of types, but we're going to focus on the three categories of honeybees. Okay, the first being the queen, which is a female, the second being the worker, which is also a female but she is sterile, and the second being a drone, who is a male but he is fertile. Okay, so the queen and the drone are both considered to be fertile. Now let's look at this particular example that we have. The queen will undergo gamete formation since it is fertile, okay, and will give rise to eggs or ova, whereas the drone will give rise to sperm. Now just for easy understanding, we have said that the queen gives rise to 2 eggs and the male or the drone gives rise to 1 sperm, but actually speaking, there are many eggs and many sperms. Okay, now what happens is the queen, when she starts laying her eggs in the brood cells of her hive, okay, so those hexagonal cells, as she starts laying her eggs, she also has the capacity of laying sperms along with those eggs. Now, those sperms were collected by mating with males before, okay, by mating with drones before. Now, let's observe what happens. Suppose if she puts an egg and a sperm into those particular hexagonal brood cells, fertilization will occur, which means I will have a diploid organism, and that's going to be a female. Now we have 2 types of females, the worker and the queen. Now depending upon what type of nutrients are fed to this existing female, you will either have a worker or a new queen. For example, if you feed it with something called as a royal jelly, you will have a queen coming in. But if you feed it with no royal jelly, so plain nutrients, so royal jelly has good nutrition, okay, you will have a sterile worker. So the nutrients help in the development of the reproductive organs of that particular female. But that's not our concern over here, because that in a way is sexual reproduction. But we are going to focus on what is really happening to that other egg cell that you can see there. Now suppose if the female goes and simply lays this particular egg cell alone in the brood cell. Now what will happen? This egg alone will give rise to a drone, which means the gamete itself can undergo a form of asexual reproduction. There is no fertilization taking place, okay, no fusion, yet that particular egg can mature to give rise to a male, which is a drone. So this is also a type of asexual reproduction, and this is what we call as parthenogenesis. Okay, so one thing that we need to take away from this is the females are always diploid, whereas the males are always haploid. Now, a further understanding of parthenogenesis you will understand in the latter videos, okay? But for now, I hope you have understood what is the concept of parthenogenesis. Now, let us take a couple of questions. Let's go through a couple of questions. The first one. If the stalk contains 46 chromosomes and the scion contains 30 chromosomes, then how many chromosomes are present in the root and the egg cell of the resultant plant, respectively? Now 46 and 30 chromosomes are the diploid numbers. So the root being a diploid cell, definitely the root should have 46 chromosomes of the stock. Whereas the egg cell, being a haploid cell, the scion is 30, which means the egg has to undergo meiosis, is formed through meiosis. So therefore, can I say 15, half the number? Okay, so therefore the answer to this particular question is A, 46 and 15. 46 in the root and 15 in the egg. Let's go to the second question. Longitudinal binary fission is found in dash, and you have four options here: Paramecium, Amoeba, Euglena, and Planaria. Now, why the word longitudinal very specifically? Now, for example, if we take Paramecium, it has got cilia all over, so it does not really matter whether I cut it transversely or longitudinally. Okay. Second one, Amoeba, irregular in shape, does not really matter how I cut it. Planaria, we don't really speak about binary fission in Planaria, we speak about regeneration in Planaria. But if we take Euglena, Euglena is biflagellated, so it is important to have longitudinal binary fission, transfer one flagella to one daughter, transfer the other flagella to the other daughter. From there, a replica can be formed. Okay, so longitudinal binary fission very specifically here is going to be seen in case of Euglena. So students, I thank you for attending today's video lecture. I hope you have understood the topic well. Kindly go through EduRev Infinity, where you will have access to a whole lot of other material. You will have access to tests and notes prepared by a lot of NEET experts, and I'm sure this is going to help you to raise your nationwide ranking in your NEET exam. Thank you.